Hey everyone, Dr. Chem here, and I am absolutely thrilled to guide you through today's explainer. We're going on a highly focused, truly cinematic journey right down into the microscopic world. Today, we're exploring a technique that sounds, well, incredibly complex, but is actually elegantly simple in its design. So get ready to shift your perspective entirely down to the molecular level. Picture a standard biological sample. To the naked eye, it might just look like a boring drop of clear liquid, right? But zoom in, and it's this chaotic, bustling soup of proteins, nucleic acids, and salts. They're all completely different shapes and sizes, just constantly bumping into one another. So how exactly do scientists sort through this invisible chaos to isolate the specific building blocks they actually need to study? That right there is the ultimate molecular sorting problem.
Section 1. Welcome to Gel Filtration, also known as Size Exclusion Chromatography.
So what is this exactly? Well, gel filtration separates molecules entirely based on their hydrodynamic radius, which is essentially just a fancy way of saying their size and shape in a fluid. It is honestly the most powerful chromatographic technique we have for getting reliable data about biomolecules under their native, natural conditions. At the very heart of this process is the gel filtration matrix. Imagine taking a chromatography column, basically a simple glass tube, and filling it with a packed bed of microscopic sponge-like beads. These little beads are absolutely riddled with internal pores and channels of varying sizes. And here is the golden rule of gel filtration in the lab, something you never forget. You must never, ever allow this packed bed to run dry. If you do, cracks and fissures will develop in the matrix, your separation is completely ruined, and yep, you have to repack the entire whole column from scratch.
Section 2. The core separation principle. A microscopic race.
Now, as the elution buffer flows down through the column, it actively pushes the molecules through. And I absolutely love using a VIP analogy for this. Think of the massive, large molecules as VIPs. They're simply way too big to fit into the microscopic pores of the beads. So what do? They take the express lane. They glide through the spaces right between the beads and travel rapidly straight down the column. But the smaller molecules? They're more like wandering tourists. They're small enough to wander into the maze of channels inside every single bead they encounter along the way.
So the crucial takeaway here is that SC is entirely counterintuitive compared to how we normally think about gravity and size. The largest, heaviest items actually finish the race first. No way around it. They take the most direct path around the matrix, they equilibrate way less, and they elude sooner. The small molecules, on the other hand, are forced to explore this endless labyrinth of pores, meaning they travel much more slowly and finish dead last.
Section 3. Inside the gel matrix, the perfect sieve.
Zooming in even closer, you can see the mechanics of this perfectly. Notice exactly how the smaller molecules are equilibrating between the internal gel pores of the stationary phase and the moving elution buffer on the outside. Meanwhile, the larger particles, they literally just breeze right past the bead. The physical design of this matrix is entirely dictating exactly what gets trapped and what flows freely.
And because biomolecules come in wildly different sizes in the real world, we have to use different matrices to match them. Each one has a highly specific fractionation range. For instance, the Sepharose S100 matrix is specifically engineered to separate globular proteins somewhere between 1 and 100 kilodaltons, which is just a microscopic measure of mass. But if you're dealing with absolute massive proteins, you jump all the way to Sepharose S400, which handles molecules up to a staggering 8,000 kilodaltons. Now, you might be wondering, what happens to the tiny molecules that fall completely below a matrix's fractionation range? Well, they actually all just elute together at the very end because they penetrate the beads with equal total efficiency. There's zero separation for them at that point.
Section 4. The protein separation race, tracking the elution.
Let's break down the timeline. At the very start of the race, the first liquid to emerge at the bottom is called the void volume. This represents the total volume of buffer that existed strictly in the empty spaces between the beads. Our VIPs, those massive proteins that bypass the matrix entirely, ride this exact wave out first. Next up, in what we call the elution volume, our medium-sized molecules that navigated just some of the larger pores start to emerge. And finally, the bed volume exits. This carries out the smallest molecules and all the excess salts that ended up taking the absolute longest route possible through the gel.
Okay, so how does this physical microscopic race translate into the data scientists actually look at every day? When you map this out on a chromatogram graph, it all clicks into place perfectly. That first peak you see, that represents the large molecules washing out in the void volume. Then, the highest concentration of the absolute smallest molecules forms that final massive peak at the end of the run. This graph is literally the heartbeat of gel filtration. It takes invisible microscopic molecular movement and turns it directly into highly readable data. But, as any scientist will tell you, the lab is never perfect. Sometimes the molecular weight peaks on our graph just do not show up where we expect them to. Why does this happen? Well, proteases might have actually degraded our target protein, physically shrinking it. Or the proteins might have non-specifically adsorbed, basically just stubbornly stuck to the column's filter. If a protein unfolds, its overall shape changes. And remember, SEC separates by shape just as much as size. Or the protein might have unexpectedly clumped together with other proteins into a totally new oligomeric state, which makes it elute much, much faster than you'd anticipate. Section 5. Powerful real-world applications. High-stakes biology. So, why does all this matter outside of a textbook? Let's explore how SIC is actively utilized for some seriously high-stakes real-world biological missions. One truly brilliant application is spin desalting, which is used all the time for rapid buffer exchange. Waiting for gravity flow columns takes forever, and it can really dilute your precious sample. Instead, scientists use a centrifuge. First, they spin the column to violently clear out all the liquid in the void volume. Then, they add their actual biological sample and spin it again. The massive centrifugal force jams the small salt molecules deep into the matrix pores, while the pure, large macromolecules shoot straight through in almost the exact same volume they started in. It is incredibly fast, and it completely eliminates sample dilution, a total game changer.
And perhaps the most cutting-edge application happening right now is the isolation of exosomes. Exosomes are these ridiculously tiny vesicles, just 30 to 150 nanometers in size, that are secreted by cells. They are literal snapshots of their parent cells, carrying important RNA and proteins, and they are absolutely vital for how cells communicate with each other. Historically, capturing them required a grueling, highly labor-intensive method called differential ultracentrifugation, or DUC. But SCC has stepped up as this highly reproducible, low-cost alternative. It yields exceptionally pure and, more importantly, functional exosomes without needing all that intense, wildly expensive specialized equipment.
Which really brings us to our final thought for today. Because exosomes provide this pristine snapshot of the cellular environment, they hold absolutely massive potential as targeted drug delivery vectors and as early biomarkers for both chronic and acute diseases. If size exclusion chromatography gives us the reproducible, pure yield we desperately need to study them at scale, well, could this elegantly simple method of sorting the invisible actually be the master key to the future of personalized medical diagnostics? It's a fascinating question, and it's exactly the kind of question scientists using this technique are answering as we speak. Thanks for joining me on this explainer today. Keep wondering and definitely keep exploring.